Hi class, my name is Danae Hall. Today is September 11th and right now I'm doing the Dilutions lab. I've got my gloves on, my goggles, my apron, and I wiped down my workstation with 10% bleach. Right here are my 6 labeled Petri dishes and my 6 labeled test tubes, and I need to pour the yeast to get that started. Alright, so I'm going to pour some alcohol into my alcohol cup that is labeled. Now I'm going to sterilize a pipette. Alright, so I don't have any more alcohol in my pipette, so I'm going to want to pipette 2¼ milliliters of distilled water, which is right here, into each one of these test tubes. So now each test tube has 2.25 milligrams of distilled water in it, and the yeast is ready. So I'm going to start with test tube 10-1 right here and add 0.25 milligrams of yeast to this test tube. Now that the yeast is in the distilled water here, I'm going to pipette it up and down to kind of mix it together really well. And then I'm going to pipette 0.25 milliliters of 10-1 into the 10-2 tube. And I'm going to pipette this one up and down to mix it around really well. I'm going to take the 10-2 and pipette it into the 10-3. And I'm going to do the same thing all the way till 10-6. Next step is to rinse the pipette in the distilled water to remove all of those organisms. I'm going to expel it into the sink. So my pipette is now completely rinsed. All the organisms are out. So I am going to move to my next step, which is to take the corresponding agar plate and the corresponding test tube, 10-1 and 10-1, and I'm going to pipette 4 drops of each test tube into the agar plate that's corresponding to it, and then mix it around so that it is covering the entire agar plate. So I'm all done with the first part of this lab. So I'm going to put my agar plates away and incubate them right side up for 12 hours, and then I'm going to invert them and then continue to incubate them for another 12 hours. And then I can dump all of the remaining yeast or S. cerevisiae solutions down the sink and wash everything else and sterilize the pipette with alcohol, and then wipe down my station with 10% bleach. Alright, so this is my incubation cupboard. So I'm going to just place all of the agar plates in here, right side up for now, and allow them to incubate for about 12 hours, and then I will come back and invert them, and then allow them to incubate for about 12 hours more. Now the cultures have been incubating for about 48 hours, so I'm going to take them out and take a look at them and count how many colonies I can see on each plate. Most of these plates have too few to count, and plate 1, this is the least diluted solution, has too many to count, but plate 2 had a good amount that I could count, and you might be able to see it here. I'll take a picture and put it in my lab report. but it has about 215, I would say. After counting all of the cultures on each plate and answering all the questions in my lab report, next is just to clean up the station and then get rid of all of the cultures. So the way that I get rid of the cultures is to soak all of them in pure bleach for about 2 hours, and then wrap them all in paper towels and seal them in a plastic bag before throwing them away. The rest of the reusable equipment needs to soak in 10% bleach for about 2 hours before it's rinsed with water and replaced in the lab kit for future use.